Hi, I'm Lauren. My presentation is about the ethics and application of gene editing. So whereas the previous presentation was about how it's delivered and the one before that was about how it actually works, mine's about how we can actually use it. So the ordering of this was really great. Ooh. Okay. Just for a bit of background, I'm Lauren. I'm a junior in high school, and I'm based in DC. And my mentor was Michael Jain. I'm really interested in this topic because a couple of years ago, I read a biography about Jennifer Doudna, who, if you didn't know, is like one of the people who was involved in the founding and invention of CRISPR technology. And one thing that the book talked about was that there's a lot of potential in this technology, but there's no regulations in place yet. Like the US government hasn't said what's acceptable, what's not. And so that was kind of what I was interested in. So my paper was kind of split into two parts. The first was how can we actually use gene editing? So I did 3 different case studies. I think each of these diseases have already been talked about, but I'll go a little bit more in depth. So the first one I did was sickle cell disease. And so this is caused by a single point beta globin gene mutation. Basically what it does is it makes red blood cells, it makes them have a funky shape, and it makes them stick to themselves. So you can kind of see in the diagram that the kind of crescent-shaped ones, they stick together, and it's bad because red blood cells have decreased oxygen transport if they're deformed, and also they have no more elasticity or plasticity. So this has a whole bunch of risks like thrombosis or blood clots, bone necrosis, acute chest syndrome, etc. So there are some existing treatments. And so this would be something like a bone marrow transplant or autologous hemiostatic stem cell transplant. But this has like a lot of problems. So only 14 to 20% of patients have HLA matched donors. And what that means is that in order to receive a bone marrow transplant, you have to have 6 factors basically match up. Otherwise, your body won't take the transplant. Even with those that have matched donors, there's a 9% risk of transplant failure and a 15% risk of graft versus host disease, which is basically your bone marrow transplant produces an immune system that attacks your own body. This is almost always fatal and generally the greatest risk when you have a bone marrow transplant. In terms of symptom management, hydroxyurea is actually pretty effective. What it does is it increases fetal hemoglobin, which is another type of hemoglobin, and it binds to the sickle hemoglobin and makes it so it can't stick to itself anymore, and so it doesn't deform. However, it's really not curative, and you have to basically take a pill or an infusion every single day or every week. So this is where gene therapy comes in. It can do a lot of the work for you and potentially cure a recently thought of as chronic condition. So there's 2 different types. I'm not going to try to pronounce them, but they kind of work the same way. In both of them, they create either fetal hemoglobin, as in the first type, or the second one is anti-sickling hemoglobin. Basically what it does is it's similar to the hydroxyurea. It binds to the sickle hemoglobin and prevents it from sickling. So in the first type, this was 93.5% successful, and there were no cases of cancer or graft versus host disease, which is a huge benefit over bone marrow transplants. However, 95% of patients had grades 3 to 4 adverse effects. So for reference, adverse effects are like side effects. They're graded on a scale from 1 to 4, 1 being mild like a headache, and 4 being like I'm going to die. And so these effects were actually pretty bad, but they probably are able to get reduced because they were attributed to the conditioning regimen patients had to go through beforehand. So patients had to go through chemotherapy to essentially wipe out their original bone marrow. And it could also be an immune reaction. Similarly, the second type, which produces anti-sickling hemoglobin, was also really successful. 3 months after treatment, there were 0 evasive occlusive events, which just means there were nothing like there was no bone necrosis, no chest syndrome, no blood clots, things like that. And this also had less adverse effects, which only 1/3 of patients had, but there was an increased risk of blood cancer. There's not enough research yet done to basically compare why one had increased risk of blood cancer and one didn't, but that's definitely something to keep in mind, especially as we look at other types of treatment. The next disease I looked at was SCID, severe combined immune deficiency syndrome. And this is basically where you have no immune system at all, and you have no T cells or B cells. This is very deadly, and again, the only treatment is a bone marrow transplant, which is pretty unsuccessful and has the same risks as I talked about before. So SCID is interesting because there are like hundreds of different gene mutations that cause it. So I only looked at 2. I looked at ADA SCID and X-linked SCID. In both of these, it corrects the faulty gene. So in the ADA one, it corrects the ADA gene and allows you to produce the ADA protein. And so in ADA SCID, this was 97% effective. This is really effective compared to any other previously known treatment. It was also really effective for older children, which is really important because a lot of SCID research is only targeted towards young children since the life expectancy is so low. But this was ADA SCID was tested on children about our age, so I'm 17, and it worked for them. This also had less side effects with only 17%. For X-linked SCID, it was also pretty successful. This was a very small clinical trial. So 7 out of 9 cases worked, but there was a huge drawback. 4 out of 9 patients developed leukemia, which is obviously less than ideal if almost half of the patients are developing cancer. So I had a couple of theories as to why X-linked SCID had cancer, but ADA SCID did not. The first is that the trial length. The X-linked SCID trial followed patients for about 10 years, so a really long time, but the ADA SCID one only followed them for 3 years. All the cancer developed in year 3 or 4 in the X-linked SCID trial. So it's possible that children treated with ADA SCID gene therapy were also developing cancer, but it just wasn't followed up on. The second way that it might have happened is that the X-linked SCID trial had longer-term T cell production. So that means that the immune system had a higher boost, it was working better. Except if your immune system is overactive and there's producing too many cells, that's one of the ways you get cancer. So it could also be potentially from that. Finally, I looked at hemophilia. Hemophilia is actually the most simple one. It's just you're missing a clotting factor, either 8 or 9. That should say 8, either 8 or 9, and that determines the type. The current treatment is recombinant clotting factors, which means you go to a hospital and you get clotting factors infused in you like once a week, once a month. This is pretty effective. However, it has one major drawback. Because you're basically adding in recombinant clotting factors, so things into your blood that aren't yours, you didn't make them, then you can develop inhibitors, which is essentially an immune reaction, and your body won't take them anymore. The idea is that with gene editing, you're producing your own clotting factors, so you won't have inhibitors. Just for the sake of time, I'm not going to go through this, but it was very effective. There were 43 fewer bleeds overall and an 80% reduction in joint bleeds. So the second part of my paper was about the ethics. Obviously, gene therapy has the potential to do great things, but only if it's equitably distributed. So if you look at the map in the top right, you can see kind of it's just a map of the United States, but all of the gene therapy trials are basically concentrated around the coast. So you think about like California has like UC Berkeley, Stanford, etc. The East Coast has most of the Ivy Leagues. The middle of the country basically has the Mayo Clinic, and that's about it. So if you live in the middle of the country, you're kind of out of luck to access gene therapy, which is a huge problem in terms of accessibility. More broadly, geographic accessibility is a huge problem as well. Wealthier countries like the US and China, so on this map, it's the countries that are darker, often have more access to gene therapy. Indeed, this is true, like the US has a robust gene therapy program, so does China, but countries and places like Africa oftentimes do not. We've actually seen this before with the COVID-19 vaccine, where wealthier countries, the US, China, Europe, got the vaccine first and then had to send it over to less wealthy countries. Obviously, we want everyone to be able to access life-saving treatment, so we need to figure out a way to make gene therapy more accessible more broadly. Ooh, why is it like that? All right. The next thing to consider is costs. So this is a graph based on my research of the cost of, quote, like normal treatment versus gene therapy, which is in red. So I couldn't find, there's no cost on SCID because it's not FDA approved yet, which is why that bar is blank. The interesting thing to note for hemophilia is that normal treatment is actually way more expensive than gene therapy. But this is kind of misleading because gene therapy is a one-time cost. So $3.5 million is a lot of money at a one-time cost. Whereas for hemophilia A, the lifetime cost is $20 million. So yeah, that's a lot more expensive, but it's spread over the cost of a lifetime. If you live for 80 years and go to the hospital like once every couple of months, once every month to get recombinant clotting factors, it's cumulative. It's not a one-time upfront cost. However, it's also relatively unclear if insurance will cover it. The general consensus is that government insurance, so Medicaid, Medicare, might cover it, but private insurance probably won't, just given that it's so expensive. The other thing to consider is that if patents expire, then genetic and biosimilars could enter the market and reduce the cost. Finally, the last thing that I looked at was eugenics. This is probably what everyone thinks of when they think of the ethics of gene editing. If I can make my child 10 feet tall or have blonde hair, blue eyes, play basketball, be super strong, then I probably would because I want what's best for my child. Unfortunately, this leads to what many people are calling, quote, designer babies. And off-label use, so like things for like eugenics, can be really dangerous and spread inequality. For example, think about the high cost of gene therapy. Remember, one treatment for hemophilia is $3.5 million. Well, if only the rich are able to access gene therapy, then people who are in poverty or living in a lower quality of like life won't be able to access it. If you have no money, you won't be able to make your kid quote unquote genetically better. Well, if you can't make your kid better and rich people can, then rich people are just going to have more inequality. They're going to have more money, have more success. That only widens inequality. The final problem is that we have a huge lack of regulations. For example, a scientist in China genetically modified a pair of twin babies to be HIV resistant. However, he was thrown in prison because there were just no regulations. Society wasn't ready for the implications of it. And the scientific community actually hated him for a long time because the scientific community just wasn't ready to determine where do you draw the line between curing a disease and eugenics. So that was basically my presentation. I was really interested in the applications of how we can use it, the potential benefits, but also the potential drawbacks and where we have to draw the line for regulation. Thank you. And if you have any questions.
Thanks so much for your presentation, Lauren. That was super interesting. And was really interesting, like you pointed out, the order of presentations and how it worked out in terms of sequence and understanding gene editing. A really well done presentation, and I like the way you kind of thought about it, not only from the mechanisms, but again the applications, clinical applications, and ethics, and all of that. So, question that I have, because I think what really stood out to me was like the SCID case studies that you kind of compared, and the difference between the two, and the fact that one followed the children for a much longer period of time than the other. Do you have more insights on like how that can happen, or why it's not a standardized kind of length of time that they're looking for this between both of those studies?
Yeah, so I think both of those, obviously this is like relatively new treatment. So I think somebody else has brought up that we don't really know the long-term effects of this yet, just given that it's such a new treatment. And so I think that the reason the ADA SCID one only followed them for a couple of years is after a couple of years, they all looked relatively stable. All the children, they were going to school regularly. They didn't have cancer. They didn't have any immune reaction. And so they appeared very stable. And it was basically like that 3-year cutoff mark that like after that 3 years, that's when a lot of children started developing cancer. And it's very unclear as to like why. Obviously the X-linked SCID was a very small study. But as up to like 3 years, they all appear very stable.
Gotcha. Thanks. And I'm going to the Q&A. I see we have a question, and this is from Stacy. She's interested in knowing a little bit more about how you research the costs of the different treatments. And are they just the cost to the consumer, and how did you calculate those costs?
Yeah. So what I did for the cost for gene therapy was what I looked at sickle cell, SCID, and hemophilia. Sickle cell and hemophilia both have, sorry, I'm trying to find the slide. They both have FDA approved treatments. So basically I took the cost from there. For sickle cell disease, there's actually a lot of research done on the cost and also the inequality caused by it. Because sickle cell disease predominantly affects like African American communities, there's a lot of research done about how the prevalence of sickle cell disease contributes to things like systemic inequality or cyclical poverty. So I kind of took numbers from there. Hemophilia was really interesting because there's actually 2 different costs. The cost if you just have recombinant clotting factors and you don't develop inhibitors, everything goes perfectly. It's actually pretty low because all you have to do is go to a hospital every month and then like receive an IV infusion. So if your insurance covers that, you're pretty much set. The problem is if you develop inhibitors, it becomes a lot more expensive because then recombinant clotting factors don't work. So you're in the hospital a lot more often, you have a lot more bleeding, you have a lot more like medical treatment that you actually have to do. So when I looked at quote like normal treatment, I looked at the cost with inhibitors because that's like the worst case scenario.
Gotcha. That makes sense. Great. And then I'd just be curious too to hear more about your interests and kind of from now doing this research, what kind of is your next direction with this, and what do you want to do next?
Yeah, so I'm really excited. This summer, I'm interning at a clinic near my house, and they do oncology. And I talked to the head of the doctor, like head doctor there. She said she potentially was interested in giving me my own project to work on. So it would be really awesome if I can use kind of like the theoretical aspects I've learned here, like how it works. Ooh, sorry. With like how it works, like how it can be applied, and maybe develop something there, or something related to what she's doing. That would be really cool.
Awesome. Congratulations on getting that this summer, and I'm sure they'll be impressed by the background that you have and your approach to things. Awesome. Thanks so much, Lauren.